Hey everyone, we're going to start into chapter 6, which is continuing our journey in talking about proteins, and now specifically looking at enzymes. So, we're going to be talking about some of the physiological significance of enzymes, the origin of their power or how they catalyze reactions, and some of those chemical mechanisms that allow them to do catalysis. In particular, we'll be looking at examples of some of those catalyses with chymotrypsin, which there is a video up right now looking at how chymotrypsin catalyzes its reaction, and also another enzyme called lysozyme. And then the last kind of half of this chapter is going to be focusing on how enzymes are inhibited and the kinetics of the enzyme. And we'll be focusing on that in the second video of this chapter. So again, what are enzymes? Enzymes are a type of protein. They are categorized as a protein that catalyzes a reaction. So remember, that's just increasing the reaction rate without actually using up the enzyme or the catalyst in general. That is the definition of a catalyst. It speeds up the rate of reaction without actually getting used up. And enzymes are just the biological type of catalyst that almost all organisms and life are going to be utilizing. Now, most of the time, enzymes are globular proteins. So remember, we talked about different types of proteins, the fibrous versus globular. Fibrous are going to be proteins that have repeating secondary structure. So they might be only made up of alpha helices or only made up of beta sheets. Globular proteins are proteins that have generally a mixture of structures, motifs, and they also can have areas that have no secondary structure. So remember, we can have those unstructured regions of globular proteins. So again, review the difference between those two types of proteins. And remember that most of the time, enzymes are going to be globular. So they have these kind of diverse regions on them. Now, there are some types of other molecules that catalyze reactions. So the example here is an RNA. RNA is not a protein. We're going to be talking more about molecules that are made up of nucleic acids. Remember, proteins are made up of amino acids. RNA, DNA, those are made up of nucleic acids. And we know that there are actually some RNAs that help catalyze reactions. So these are a special type of enzyme in that they are not a protein, but they do catalyze a reaction. Now, enzymes in general, as a study in the field of biochemistry, is one of the oldest areas of study. We've been looking at processes of enzymes and kind of isolating enzymes and looking at what they do all the way back into the late 1700s. And most of biochemistry in the early coming of age of the study of biochemistry was just dominated by the study of enzymes. And still to this day, we're still finding and classifying enzymes and figuring out their mechanism of action. So why use an enzyme? Why use a biocatalyst, a catalyst that is biologically made out of a protein, over a catalyst that is inorganic? So we know that there are tons of other catalysts that are inorganic in nature, and you probably have already used some of these in, for example, organic chemistry. So why use a protein, a big globular protein, as a catalyst instead of, for example, something like molybdenum or graphene? These are all inorganic catalysts. Well, one of the major reasons in an organism you would want to use a biocatalyst is you have greater reaction specificity. a lot of these inorganic catalysts can work on numerous types of reactants to make all kinds of different products. So they speed up general reaction types, but they don't exactly specify only one single chemical that they will react. Proteins and enzymes can do that. They have specificity for specific molecules, for only one particular molecule in some cases, or maybe only a class of 3 or 4 molecules. And so this is perfect for a biological organism where you don't want to have cross reactions and you need specificity per molecule. Biocatalysts are also great at conducting their catalysis in normal reactive conditions of about pH of 7 and at 37 degrees. No surprise here, organisms have evolved in these conditions, and so it would make sense that the enzymes that have evolved are also great at conducting their enzymatic reactions within these pH levels. Biocatalysts also have higher reaction rates. So in terms of a biological organism, that is very good in the time frame that we're looking at. We need something that is going to be able to take our particular specific reactant and quickly, very, very, very quickly turn it into product. And then one really, really, really key thing is biocatalysts have a capacity to be regulated. Again, inorganic catalysts, simple inorganic molecules, they don't have specificity. They're very uncontrolled. There's no real way to turn them on or off. You add them in, they catalyze the reaction. They can catalyze lots of different reactions. Biocatalysts and enzymes have the ability to be turned on and off. That's what I mean by capacity for regulation. And in an organism, again, this is a good thing because sometimes we do need to turn pathways on and off by essentially regulating these enzymes. So for example, one very classic type of pathway that we'll look at later, and if you go into Biochem 2, you'll see a lot, is your metabolism and regulation of glucose. So a lot of people know about glucose and insulin regulation. And depending on how much glucose you take in, how much you eat, right, you want to regulate the levels of insulin that you are producing. This is regulated through enzymes, and we have the ability to turn on the production of insulin and the use of insulin, and also turn off the use and production of insulin. And that is very useful depending on the environment that the organism and the state of the organism that it happens to be in, right? If you have a lot of glucose versus a little bit of glucose, you want to have your insulin levels high or low, right? So this capacity for regulation, the ability to have some region on the protein, on the enzyme that we can help turn it on or off, is very important for biological pathways. So again, enzymes we know have very specific substrate selectivity. Here's an example where we're looking at 3 different compounds that has the ability to go into an enzyme, which is phenylalanine hydrolase. And you can see these compounds have similar structure, but the only specific structure here that will bind to phenylalanine hydrolase will actually make product. So sometimes we can have binding of similar structures, but no reaction, meaning that the enzyme might kind of bind something that looks similar to its substrate, but it can't actually drive the reaction. And so catalysis won't occur in that sense. Or you just straight up won't have binding. As in this case right here, we have no binding of the actual original reactant. But when you do have the very specific actual substrate, you will have binding and reaction and catalysis of the reaction. And I want to note here, so this arrow is a little off, but here we have, this is the binding nature of the enzyme. And if you look at these two molecules right here, they are very similar. In fact, the only difference between them is that they are enantiomers of each other. So remember, enantiomers, the R versus S, they are mirror images of each other. So enzymes can and do even specify between mirror image molecules. So molecules that have the exact same chemical formula, they have all of the things bound to the same place, but in a different arrangement. So again, these have all the same structure except that they are mirror images, and binding pockets in an enzyme can even distinguish to that point of selectivity. This is why we talk a lot about things like the rotation of specific amino acids and other types of complexes that biological organisms utilize. So a lot of vitamins, for example, and other small molecules, they will have two different mirror images. They'll have the R and S formation. And enzymes can and do distinguish which one of those they will bind and catalyze. So here's an example of an enzyme and substrate in a complex. Enzymes, again, are just proteins, usually globular, and they can have one or more what we call active sites. These are the sites in which they bind a substrate. Remember, we've talked about the word substrate. You've also probably heard this called a ligand. So this is the binding site or the active site, sometimes it's called the active or the binding site. And substrate, we also sometimes call the ligand, right? Ligand is just something that binds to a protein. And specifically, we'll talk about substrates, which are ligands that are binding into enzymes. Now, remember, you can have proteins that bind to a ligand but are not enzymes. And one of the best examples of this that we talked about previously is something like myoglobin. Myoglobin is a carrier protein. It binds onto oxygen, it holds onto oxygen. So it has a ligand, it has a substrate, it has something that it actually binds, but there is no reaction that is happening. It's just carrying it. So it's just holding on to it and it will let it go later. This is just an example of a carrier protein and not an enzyme. Remember, enzymes have to actually catalyze a reaction. There's some type of change of reactant to product. So these binding sites, they have key amino acids that are going to help to hold on to and conform to whatever the ligand or substrate is that the enzyme is driving the reaction of.